Hey everybody, Mr. Murphy here. I'm going to talk about the statistical tests that we can run on our photosynthesis lab. Looking at this screen here, this is an Excel spreadsheet that I have shared to you through our calendar. We are like real science people. We're using Excel and not just Google Sheets. I suspect Google could do all of this, but I already have it set up in terms of creating the graph and things in Excel. And so I think that's going to be the easiest for us to just plug our data in and get what we need. But on the first tab of this is the chart, which I've shown previously in class. I really do think this is a super helpful thing. I never had anything like this, and statistics was often a mystery to me even through my college years in terms of how I was applying things, and I'm sure I did it horribly wrong for many, many years. But this is nice as it relates to biology, because this helps us to look into exactly what kind of data do we have in terms of our independent and dependent variables, and it sets us up for success in terms of knowing how we can meaningfully analyze that data. So looking at this, is it a continuous value or a discrete one? So a discrete one would be like counts of something where it has to be 1, 2, 3. Whereas continuous data could be 1.01, 1.003865. It could be any infinite number of decimal places. So in this experiment, we do have continuous dependent variables because all of us used a time as our dependent variable, the time for the leaf discs to rise. So we're over here on this side. And then from there, it's asking, is my independent variable continuous? Now this depends. If you set up perhaps like a control and experimental group in terms of changing the pH or the temperature or the type of light or something along those lines, then you do not have a continuous independent variable. But maybe you are changing the concentration of carbon dioxide or the bicarbonate in the solution, or maybe you were changing the distance from the light or something like that, where you could have like a gradual step-by-step change, then you might have a continuous variable. If that's the case, then we'll follow the path here. Do I really have two dependent variables? No, because we're probably measuring only the time, and otherwise we are sort of in control of what the independent variable is. This path would be like if we were doing something ecological, we were out in a field, actually studying something where we're looking at the relationship of two things that aren't really within our control, then we would be doing this. Anyway, so in that case, we'll be doing a linear regression. So that's going to be the first one that I talk about, but I do want to just be clear about what the other path is that for this experiment we may have followed, and that is that maybe you were doing like just two different groups. Hot and a cold, or room temperature and warm, or whatever the case might be. In that case, where you had two setups and you were just repeatedly trying them and getting multiple data points for that, then you would follow this no path here. Is my dependent variable paired? Likely no, because a paired test would be like if I tested these leaf discs now and then a week from now tested them again, that would be paired because I'm looking at the same leaf discs. So the value that these 10 leaf discs got today is linked, paired to the value that those same 10 leaf discs got on another day. So that's not the experiment we did, but that's how you would end up there. So we're over here. Is my independent variable greater than two categories? So for this experiment, I limited you to just looking at two things, but there would be a statistical test you could do if you did, hot, cold, and room temperature, for instance, or you did multiple groups like that. Then you could actually statistically analyze those all at once. So we're going to end up here with the student t-test, which was one of the statistical tests we had done on the summer work. So linear regression, student t-tests. Let me talk about it. For linear regression, you'll go to the regression tab down at the bottom, and it's all set up for you. So it's ready to go. You just need to drop in your data into the independent and dependent columns. When you do that, please rename it. So for instance, my dependent variable is the time, and I'm going to put in parentheses S for seconds. I recommend using seconds as your time, so just convert it to seconds, right? Because you can't have like 4 minutes, 23 seconds, and then like do a statistical test with those numbers, because 4M23S doesn't really mean anything. Just convert it to seconds. And then for the independent variable, I just have some gradually increasing thing. So I'm going to just say that this is the amount of poison in milliliters that I've added to my solution. Which then, when we just kind of casually look at this, we see as we increase the amount of poison, the time for the leaf discs to rise has increased. The time you should be using is the time for half of the leaf discs that did rise to rise, if that makes sense. So if I had 10 go up, then I'm looking at the time at which the fifth one rose. If I only got 8 to go up, then I'm looking at the time for the fourth one to rise. If you get an odd number that rose, then go between. So for instance, if I had 9, I would look at the time directly in the middle between the fourth and fifth disc, and I would use that value. If you have questions on that, just let me know. I already just made up data and plugged it in. Just note that if I change these numbers, my graph does change. So you will not have to do anything in terms of graphing. You just plug those numbers in, and it should automatically update the graph for you. Now, something you'll want to do is change some things about the graph, and that's important because don't just copy and paste this graph straight into your lab report, but instead go in here, change this to actually be your independent variable. So I would say, poison, parentheses, milliliters. I would change this to be time, parentheses, seconds, and then I would give it a title. Now, the title could include the words linear regression, but, linear regression of time versus poison or whatever. So give it a meaningful title. You also notice that it gives you the equation for the line that is formed here and the relationship. The point of this test, I probably should have started there, is to determine whether there is a meaningful relationship between the two variables. And so as we gradually increase the independent variable in this case, we're looking for basically, first off, was there like a trend of the dependent variable changing? In this case, yeah, I can see that trend increases over time. But then the next question we need to ask is it enough of a trend to really give us statistical confidence? Because you can plug any numbers in here, and yeah, sure, maybe when the graph is spaced out just right, it looks like it's an increase, it looks like a decrease, but it's always going to. The real question is the data in line enough to give us a compelling support mathematically that yes, there is in fact this positive correlation between the time and the amount of poison, and that's where this bottom part comes in terms of the actual linear regression. Something you'll want to do is there's a little box here which gives you an instruction. This is going to come to you blank. And the reason for that is because these cells have to populate all at once when conducting the statistical analysis on your data. And so what I included in this little box is that you're going to click on G27, which is right here, this one next to slope, and you're going to paste this exact line. So I'm just going to copy this, and I'm going to go here and just paste that in. If you need to at this point, maybe you had, maybe you had one fewer data point, or maybe you had 3 extra data points, whatever it is. But make sure you move these columns. You can grab the bottom little box thing and you can move those up and down so that you are capturing your full data set, and do that before you hit enter, because once you hit enter, as you'll see, all of these cells populate and give us all the statistical information that we need for this statistical test, linear regression. Now, what do all these numbers mean? They mean a lot of things. And as you, hopefully venture into statistics a little bit more in your educational career, you'll learn a little bit more about the specific relevance of these different things. Some things like slope and intercept should hopefully make sense to us from some, basic math principles. But the two that we're really going to be looking at, or actually the 3 things we're going to be looking at, are the R squared value, the F value, which is our actual test statistic, and then the degrees of freedom. So the in the case of actually finding out does this mean anything, we need a table similar to our chi-square table, similar to the t-test table that we saw in the summer work. And so for us, in this experiment, we're going to be looking at the alpha 2, which is a two-tailed test. It's a two-tailed test because I don't have an expectation of whether this is higher or lower. I'm looking for basically, is there a relationship, period, either direction, going higher or lower. There's more information to learn about, one-tailed versus two-tailed and what kind of experiments would be one versus the other, but we're not going to dive into that today. And what I'm going to do is look at my test statistic, which is 36.31. This is kind of like my chi-square value, is how you can think about it. It's your chi-square value or it's your t-value. And then I'm going to look at my degrees of freedom, which is 8. If I go over to this table, and sorry, I'm going to move this because I know that I'm in front of it. You can disregard the table on the right here. I'm looking at this guy, and let me make this a little bigger. Okay, hopefully you can read this on the video. Anyway, I'm going to look at the two-tailed test, and I'm looking for that 5% mark, because if my F value exceeds the value in this column for the correct number of degrees of freedom, then that tells me that there is a less than 5% chance of this outcome occurring due to randomness alone. And that would tell me that I could have confidence that, yes, my variable is actually influencing the dependent variable. My independent is statistically significant. So looking at that 5% column, I said I had 8 degrees of freedom, so I'm looking for 7.57. So if my F value is greater than 7.57, then we are good. It is statistically significant. And sure enough, mine is 36.31. Now, I benefit from the fact that I made up the data, so I made it statistically significant. You should not do that. Just go with what your data is. And if it's not, great. You have something to talk about, that the data did not support the hypothesis, that we did not reject the null hypothesis of our statistical test, and then you can discuss why that might be, or what aspects of the experiment should maybe be tweaked to better capture the data, what might be done. Or think about it some more, and maybe you can demonstrate that, oh, there was a flaw in our underlying thinking, and of course it wasn't going to influence it, or whatever. So it just depends on what your experiment is. It depends on what you saw, and it depends on what you're thinking about the relationship between your variables. So don't be swayed or don't be thrown off by data that doesn't, reject your null hypothesis, that doesn't show statistical significance. Because that's science, and that's most of what science is, and it's okay to not have data come out statistically significant. That doesn't mean failure. It may mean that you need a closer look at your lab procedures and how you conducted the experiment, but we learn things no matter what, and so that's fine. And so this 36.31, I would say, yes, it exceeds the 7.57, and therefore I can state that this is statistically significant results, and I can also say that I reject my null hypothesis, and that your null hypothesis for an experiment like this was probably that there is no relationship between the two variables, and your alternative hypothesis is that, yes, there exists a relationship between these two. That third data point I mentioned, the R squared, is actually a percentage relationship between these two things. And what I mean by that is how much, based upon how compelling your data is, how much of the change in the dependent variable can be attributed to the independent variable. And so I have 0.82, which is 82%, which is pretty darn high. So that just gives you another sort of marker to say that, well, there's the 82% of the trend of the dependent variable, my time going up, is related to the independent variable that I was manipulating. Okay, so that's this experiment. You'll just drop in your own data. Don't forget that you will have to drop in the equation down here, but just follow the instructions, and certainly ask me for help at any time, and I can do that. In fact, if you are going to email me at any point, don't expect an immediate response if you email me on like Christmas Day, but send me the Excel file that you're currently working on, like just attach it and say, hey, could you take a look at this or whatever. That just might make it easier and have fewer back and forth emails if there's something that you do want me to take a look at. All right, that's regression. For groups who are going to be conducting a 2-sample t-test, that is our student t-test. If you're conducting the t-test, click on the 2-sample t-test tab down here. Now this just has random data in it right now, so you'll replace this with your own. If you have 10 trials that you did, great. Just drop that. The rest of these, you can just delete them. That's fine. And then for each of these, I know you would have done 10 maybe control vials and 10 regular ones. If you did fewer than that, that's okay. Just include as many as you did. A t-test really is most valuable when we have a larger sample size, but in the context of this experiment, we do the best we can. So just drop in whatever you happen to have. You don't have to do anything fancy. So if I had 10, then I just put my data in here. I did 10 control vials. I did 10 treatment vials. Label those better. So if you were doing, for instance, type of light. So if I did lamp versus sunlight, I don't think any groups did that, but you know what I mean. Just label those so that way they are here. And then it does two things. It gives us a nice graph here. I'm going to bring it back to my full data. And it also gives us the statistical information down here. It gives us some just basically reference statistics about our data. And it also gives me the student t-test data. Thinking back to the summer work, we did a t-test like this. And these three data points right here are the important information that we need. So the first one is the t-value, which is kind of like your chi-square value. It's what you're actually going to use to reference the big table here. We have our degrees of freedom, which is based on the total number of observations, like -2 or something like that. And then we have our p-value. The p-value is actually just sort of the answer. The p-value is basically what is the percentage chance of getting these results purely due to randomness alone. And so right away I can see that I have a statistically significant result because it's 0.013, which means there's a 1.3% chance of getting these results due to chance alone, and our cutoff is 5%. So as long as I'm less than 0.05, which would be 5%, then we're in the clear. That means I have statistically significant data. But I do want to just go over real quick how to look that up on the table yourself. So if I look at the table here, I'm going to zoom in a little bit. Sorry. Okay, so we're looking at the alpha 2, which is a two-tailed test because our values could have gone up or down. As I mentioned on the regression one, there's more to one-tailed versus two-tailed, but we're not going to get into that right now. And I'm looking at the 5% cutoff, which would be that there is less than 5%. I want to be to the right of this. So there's less than 5% chance that this was due to randomness alone. I had 46, I believe, as my... So I'm looking at the third column, that's the 5% one, and I had 46 degrees of freedom. So I come down here and it's 2.013. As long as my t-value is greater than 2.013, then we are in the clear, and we can see that it is. It's just a smidge higher than that, which tells me that there is a statistically significant difference between my two treatments here. One additional thing, and I mentioned this, excuse me, I can't talk tonight. I mentioned this on the regression tab as well. Spruce up this this graph. You don't need to make it fancy with like colors and fun fonts and things. Don't do that. But I just mean give it a real title. Describe the two treatments that are being compared. Over here where it says number, please put your what the dependent variable was. So probably time in seconds. And then having this little parentheses, mean plus or minus 95% confidence interval, that's fine. That's just explaining what these error bars are. So these error bars are looking at basically where we have a 95% confidence that the mean actually falls is somewhere between these error bars for both of these. And that's important because, you might have two bars that are off from each other, but maybe their error bars have significant overlap. So you wouldn't be surprised then to find that your data was not statistically significant because even though you had a difference in the means, we had so much variance to our data that we don't have really confidence that those means are different. So if you get a circumstance like that where you have a big overlap of the error bars and you find that your p-value down here is greater than 0.05, that's okay. It's not a problem if we don't have statistically significant results. It's just more things that we can talk about in the lab. So spruce this up, put your whatever your two treatments were. Control and let me see if this. Oh, cool. Actually, as long as you change the thing here to I don't know, what did I say? Sunlight or whatever. As long as you change that, you'll notice that the column titles actually change as well. So I've got that set up for you, and should be good. And if you have questions, email me. Please attach this file to the email. I'll get back to you. Yeah, we'll do it, and hopefully with what we talk about in class, you'll understand a little bit more about why this is a valuable thing to do and what it means for the data we collected. And I hope we get some statistically significant results. And if we don't, it's okay. It's okay. It's part of science. That's most of what science is, honestly, is trying things, figuring out that maybe that's not the right answer, or figuring out that maybe I need to rethink my procedures to better collect the data necessary to support the claim we're trying to make. Yeah, so should be good. Email me questions, and good luck. Thanks.